This is part 2 of the Chapter 6 recorded lectures. This is where we left off, but I would like to go back. Honestly, the slides are not organized to me, they're not organized very well, but this is the first time I've used these slides, and so I just want to make sure that everyone understands what we're talking about here. Right now, we're talking about cellular respiration, but this chapter covers both photosynthesis and cellular respiration. But it's trying to introduce to you, in the beginning, it's trying to introduce to you basically how energy in the form of ATP is produced. So we started out with learning that one of the ways ATP is produced is by breaking down glucose, because within the bonds, the chemical bonds that form the molecule of glucose, energy is stored. And when glucose is broken down slowly during cellular respiration, that energy is used to produce ATP. But the ATP, and this pathway, by the way, we will come back to, but this is just showing you the two membranes of the mitochondrion, the outer membrane and the inner membrane. And the processes that we can see here are the citric acid cycle, which we will learn about later, and the electron transport chain actually occurs on the inner membrane, which we call cristae, the little folded inner membrane. You see how it's arranged in little folds. So we will come back to this when we cover cellular respiration in detail. But right now, some of the basics you need to understand are that redox reactions are occurring in the chemical reactions that we study in this chapter. And redox reactions mean oxidation reduction reactions. One molecule is oxidized, and to be oxidized, oxidation is loss of electrons. So when a molecule is oxidized, it typically loses hydrogen atoms because the electrons are within the hydrogen atoms. The hydrogen atom carries a hydrogen ion that has a positive charge, and it also carries an electron with a negative charge. So together, those particles make up a hydrogen atom. So anytime you see a molecule, such as this molecule here, C4H10, that contains a lot of hydrogen atoms, it is capable of reducing another molecule, because reduction is is gain of electrons. I apologize. Reduction is gain of electrons. So C4H10 is capable of reducing oxygen, and by converting it to water. So C4H10 is called a reducing agent because it's able to give oxygen electrons in the form of hydrogen atoms. So we say that the C4H10 is oxidized and the oxygen molecule is reduced, because oxidation is loss of electrons and reduction is gain of electrons. But what I'm trying to get you to understand is you don't see electrons in chemical equations. So you have to learn to recognize hydrogen atoms. Anytime you see hydrogen atoms in a chemical such as glucose or C4H10, when a chemical has hydrogen atoms attached, also the coenzymes NADH and FADH2 contain hydrogen. They are carriers of hydrogen atoms, which means that they are able to donate hydrogen ions and electrons to other chemicals. That means that they are reducing agents. They're able to donate electrons. And you can see here that when glucose is converted into carbon dioxide during cellular respiration, the glucose is oxidized because it loses its hydrogen atoms, meaning that it loses electrons. And here's a picture of one of our electron carriers, which is NADH, and the other is FADH2. These electron carriers are produced during cellular respiration, and they are used to make ATP. ATP is the energy currency for cells. One way that ATP is produced, I didn't really like this because it really should have been shown in the opposite direction, and I didn't mean to scribble all over the slide, but substrate level phosphorylation is when a chemical, like our green spiral chemical here, some chemical called the substrate has a phosphate attached to it, and it donates that phosphate to ADP, and that makes ADP ATP, adenosine triphosphate. So that is called substrate level phosphorylation. And then you can see, since the ADP has taken up that extra phosphate group, the substrate is missing the phosphate because it gave it to the ADP molecule to produce ATP. Anyway, this is called substrate level phosphorylation. Basically, the phosphate group just comes from some chemical that has a phosphate group attached to it. But 90% of the ATP is generated in a process called not substrate level phosphorylation, but oxidative phosphorylation. And that goes back to our picture here. What we're looking at is oxidative phosphorylation. It involves hydrogen ions and electrons that come from NADH and FADH2. So it says 90% of the ATP generated during glucose catabolism or cellular respiration is derived from chemiosmosis, which takes place in the mitochondria within a eukaryotic cell or the plasma membrane of a prokaryotic cell. The process uses oxygen, so it is known as oxidative phosphorylation, and it's going to involve an electron transport chain, going to involve an enzyme called ATP synthase, and it's going to involve hydrogen ions and electrons. Okay, so oxidative phosphorylation, it says that electrons from glucose are transferred to small molecules known as electron carriers. These electron carriers are the ones I told you about, NADH and FADH2. So in the beginning, when glucose is broken down in the first few steps of cellular respiration, some of that energy is used to make ATP by substrate level phosphorylation, but it's just a very small amount of ATP. But a lot of the energy from the glucose is used to make these electron carriers, NADH and FADH2. They are going to go on to the last step of cellular respiration called oxidative phosphorylation, and they are going to be used to produce 90% of the ATP. So the electron carriers take the electrons, which are found within their hydrogen atoms, to a group of proteins in the inner membrane of the mitochondria. And this group of proteins is called the electron transport chain. And as these proteins pass the electrons from one protein to the next, as the electrons move through the electron transport chain, every time they move to another molecule, every time they're passed down, they lose, they go from higher energy to lower energy. So and eventually they attach to oxygen along with hydrogen ions and form water. So oxygen, we call oxygen the final electron acceptor. The final electron acceptor. Energy from the electron transport chain is used to produce ATP by an enzyme known as ATP synthase. And since this process requires oxygen, it's known as aerobic respiration. Aerobic respiration. Okay, so now we're going to talk about photosynthesis, which is basically the reverse of cellular respiration. The overall equation for photosynthesis is shown here in words and chemicals. I would like for you to know both. In words, I said so, I'm sorry. The overall equation for photosynthesis. Okay, So now we're talking about photosynthesis. We have to kind of change gears. Um, in words, the equation is carbon dioxide plus water. So we know that plants take in water through their roots, and they take in carbon dioxide from the air, and the carbon dioxide enters their leaves through stomata, little pores in their leaves, little pores on the underside of the leaf. So carbon dioxide and water are converted in the leaves of plants into simple sugars like glucose. This particular one is glucose, plus oxygen gas. We know that plants produce oxygen gas, and that's how we get the oxygen in the air that we breathe. The chemical formulas are 6CO2 plus 6H2O yields, in the presence of sunlight, C6H12O6 plus 6O2. So that is the overall equation for photosynthesis. Here you see a tree, and it's representing where the reactants and products come from and where they are produced. So water is absorbed into the roots of the plants. Carbon dioxide is taken in by the leaves. Sunlight is absorbed by the pigments in the leaves. And then the leaves use the carbon dioxide and the water to make sugar, which is the food for the plant, and oxygen gas, which is released into the atmosphere. Now, organisms can be photoautotrophs, or they can be heterotrophs, and then we will learn in Bio 112 there are actually other types of nutrition as well. But we're in this chapter, we're just going to concentrate on photoautotrophs. Means that they carry out photosynthesis. They use sunlight energy to make their own food. Autotrophic means they make their own food instead of consuming it. Heterotrophs would be like animals and fungi, which consume food from their environment. So the photoautotrophs include plants, algae, and cyanobacteria, which carry out photosynthesis. As far as photosynthesis in the leaf, the process of photosynthesis occurs in the middle layer of a leaf called the mesophyll tissue. Stoma or stomata, stomata is the plural form, stoma is the singular form, are pores on the underside of the leaf, which allow for carbon dioxide to enter and oxygen to leave. It says the stomata are typically located on the underside of the leaf, and that helps to minimize water loss because water vapor also leaves through the stomata. The stomata are able to regulate. They can conserve water if they close, so they're able to close and open to conserve water. But basically, the stomata are the pores where carbon dioxide enters and oxygen leaves. And this is a chloroplast, which is the organelle inside of plants and algae, in which photosynthesis takes place. The stacks of thylakoids contain the pigment. They contain chlorophyll. The thylakoid membranes contain chlorophyll, the green pigment, and that is where the light reactions of photosynthesis take place. And then the stroma is the fluid surrounding the thylakoids, and that is where the Calvin cycle reactions take place. And those reactions do not require light. A stack of thylakoids is called a granum. Light energy can be described in as energy wavelengths, and it also has a particle nature. But the wavelength is the length from, whenever you have any energy that travels in waves, it's the length from one crest to another crest. So that's the wavelength. The longer the wavelength is, the lower the amount of energy that particular light has. And then the shorter the wavelength, the greater the energy. And here we see the entire electromagnetic spectrum, and we're mainly just concerned with the visible portion of the spectrum. And you can see the colors of